Hey everyone, welcome to Exam 1 review for Talaro's Foundations in Microbiology 12th edition by Chess. This exam review is going to cover a few chapters, chapters 1, 4, 5, and 6. So let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, welcome back. Let's go ahead and start with chapter 1 of our exam review. And just my normal disclaimer, please make sure you study all the material for the exam. These exam reviews are not, in the place of studying and, learning all the material from the chapters and from those handouts, specifically from the handouts and the lecture videos. I try to remember important topics to study for the exam, so to prepare you the best I can, but this is not a substitute for studying for the exam. So with that disclaimer out of the way, let's go ahead and start with chapter 1. Okay, chapter 1, we're talking about the different organisms and small infectious agents, and 100% you should know that these are the living organisms that are studied by microbiologists on the left, and these are not living organisms on the right. You should know that viruses are called small infectious agents. They are made up of nucleic acid and a capsid coat, right? That's called the nucleocapsid. And prions are entirely made up of proteins. So these are not alive. These are small infectious agents. And you should know that the bacteria and the archaea are prokaryotic. These are prokaryotic creatures, and the rest of these, the fungi, protozoa, algae, and helminths, these are all eukaryotic organisms. You should definitely know the difference between prokaryotes and eukaryotes. Remember that prokaryotes are unicellular organisms that lack a nucleus and lack all the other membrane-bound organelles as well. Whereas eukaryotes are either unicellular or multicellular, and they have a nucleus as well as the other membrane-bound organelles. And you should know, again, that viruses are not living organisms. They are acellular, small infectious agents. You should remember the resolution limit of the eye. We've spent quite a bit of time talking about it, so I want you to remember that for the exam. Oh, hey, Wicket. Wicket's come to say hi, everybody. Hi, Wicket. So the resolution limit of the eye, you should know that it's either going to be expressed in millimeters, so it's 0.2 millimeters, or that's the same way as saying 200 micrometers, okay? You should know that that's the resolution limit of the eye. And then you should know that microorganisms are smaller than that, right? Microorganisms are smaller than the resolution limit of the eye, so they are microscopic, right? And you should know that eukaryotic cells are typically larger than prokaryotic cells, followed by the much, much smaller viruses. And remember this, you should know that you can only see cells with a light microscope. You know, the compound light microscope from the lab. That's known as a microscopic organism. We can see organisms. We can see cells. Anything subcellular, anything smaller than a cell is considered ultra microscopic, and so you cannot see it with a normal light microscope. And what do you need? Can you beat Wicket, who just left, by the way? That's right, Wicket. You need an electron microscope to see viruses and other ultra microscopic particles, right? Does that make sense? So make sure you understand that as well for the exam. All right, and that brings us to our first concept check. By the way, these concept checks might appear on the exam, so they're worth studying, okay? So make sure you actually learn the concepts here. Which of the following does not describe a fungus? Remember, fungi they do have a nucleus, okay, because they are eukaryotes. They have ADS ribosomes because all eukaryotes have ADS ribosomes. They are decomposers, because that's what fungi are known for. Remember, fungi are saprobes, which means that they break down organic matter, like dead organic matter. They decompose. But fungi are never photosynthetic. So the answer is photosynthetic. Also remember that a parasite is an organism that becomes nutritionally dependent on a host. Remember that pathogens are organisms that do harm. They cause disease. Remember that it is in lower income countries where communicable diseases crack the top 10 in terms of deaths. However, in high income countries, usually it's other diseases such as cancers or heart diseases. You should remember the concept of spontaneous generation, this false belief that organisms come out of nothing. And you should remember that it was Louis Pasteur who disproved spontaneous generation. You should remember that. And by the way, you should try to remember what all the different pioneers of microbiology discovered. So remember that Anthony van Leeuwenhoek, how he contributed to the field of microscopy by developing this pinhole microscope, and he was able to see some of the earliest microorganisms that were depicted. You should know that a theory is not just a hunch, it is a shown explanation of why things happen. So again, concept check number 3, a scientific theory has little or no evidence to support it. That's obviously false. You should know that it was Dr. Jenner who discovered vaccination. You should know the term sterility. To be sterile is to eliminate all life forms, including endospores, viruses, and prions. You should know all the contributions of these early pioneers. Dr. Oliver Wendell Holmes, who observed that mothers of home births had fewer infections than those who gave birth in the hospital. Dr. Ignaz Semmelweis, who introduced and championed hand washing. Dr. Lister, who introduced aseptic technique in the surgical setting, right? Remember, he was the first to use chemicals to disinfect his surgical instruments. You should remember that germ theory was brought to you by Louis Pasteur and Robert Koch. You should know that Koch was the one who developed 4 postulates to link a particular germ to a particular disease. And by the way, try to remember the other contributions of Pasteur as well. Not only did he disprove spontaneous generation and showed the germ theory of disease, but he also showed how fermentation works. He developed pasteurization. You should remember that taxonomy involves organizing, classifying, and naming things. You should know the order of taxonomic hierarchy from domain all the way to species, right? So, for instance, if I said domain, kingdom, phylum, what comes next, right? You should know the next one is class, right? So you should know all the different levels of taxonomy of this hierarchy in order. So again, remember, if an organism is in the same family, must it also be in the same class? Remember, class comes before family. So if they're in the same family, then yes, they must have to be in the same class. So that's true. And you should definitely know binomial nomenclature. So for instance, the binomial nomenclature involves a 2-word epithet with the first word being capitalized and referring to the genus of the organism, and the second word being lowercase and referring to the species of the organism. And then remember, the whole thing has to either be italicized or underlined. And obviously, you should know the 3 domains of life: bacteria, archaea, and eukarya. Also, and I'll reiterate this in the next chapter, but do you remember something very important about the 3 domains of life? It was the archaea and the eukarya that were more closely related to one another than either is to bacteria. So that was something that kind of is unexpected, right? Because you would think that bacteria and archaea would be most closely related to one another, and that's because they're both prokaryotes. However, when you're looking at their phylogeny, that turns out that we share a more common ancestor, a more recent ancestor with archaea than we do with bacteria, making eukaryotes and archaea more closely related than either is to bacteria. You may want to remember that. All right, and that about wraps it up for chapter 1. Let's go ahead and dive right into chapter 4. All right, chapter 4, a survey of prokaryotic cells and microorganisms. You should definitely know what's on this slide. This is a slide explaining the characteristics of cells. You should know that all cells possess a cytoplasm. All cells possess membrane. Remember the membranes called the cytoplasmic membrane, the plasma membrane, or the cell membrane. And then all cells have at least one DNA chromosome. And then also that all cells have ribosomes. You should definitely remember that the eukaryotic cells include members of animals, plants, fungi, protists. And do you remember that the protists include both the protozoa and the algae? You should know that eukaryotes possess all these membrane-bound organelles, and the main one is called the nucleus. And you should know that prokaryotes include the bacteria and the archaea, and they have no membrane-bound organelles, and especially not the main one, the nucleus. You should know the characteristics of life. These are the characteristics of living things. I could easily see myself asking, which of the following is not a characteristic of life or a common characteristic of life? All right, and then please remember the parts of a flagella. Remember the filament, the hook, and the basal body. And remember that the filament is made up of a protein called flagellin. You should definitely know that the bacterial flagella rotates 360 degrees like a propeller, okay? It's the eukaryotic flagella that whip back and forth or beat back and forth like a tadpole. You should know the different types of flagellar arrangements. Remember, atrichous means no flagellum, monotrichous means a single flagellum, lopotrichous means a bunch or a tuft, amphitrichous means flagella at two ends of the cell, and peritrichous means flagella dispersed over the surface of the cell. You should remember taxis. Remember, taxis means a directed cell movement. Chemotaxis means towards or away from some kind of chemical. Positive chemotaxis means towards a chemical. Negative chemotaxis means away from a chemical. Remember, positive chemotaxis moves towards an attractant, whereas negative chemotaxis moves away from a repellent. Then you have phototaxis, which is directed movement towards light. You should also remember this for the exam for sure. Counterclockwise rotation of the prokaryotic flagella results in a run, right? A linear run, whereas clockwise results in a tumble. And then do you remember the periplasmic flagella? You should know it's also known as an axial filament, and this wraps around the body of a spirochete or a spiral-shaped bacterium, and it results in corkscrew motility. Now again, concept check number 1. Remember, these concept checks may appear on the exam. Which of the following best describes the action of a prokaryotic flagellum? Obviously, it rotates, so it has to be C. Next, you should know about the fimbriae. You should know that fimbriae are fine proteinaceous hair-like bristles that aid in adhesion. You should know that their job is in adhesion to cells and other surfaces. And you should know about the sex pilus. The sex pilus is formed in order to transmit DNA from the donor cell to the recipient cell. Next, you should definitely know about the slime layer versus the capsule. You should know that the slime layer is loosely organized and loosely associated with the cell membrane, whereas the capsule is tightly associated with the cell, okay? And it's more organized and tightly associated with the cell. Next, we should definitely know the components and the structure of the gram-positive bacteria versus the gram-negative bacteria in terms of the cell wall. You should know that the cell wall of bacteria is made up of peptidoglycan, and peptidoglycan is unique to bacteria, right? You do not find peptidoglycan in archaea. You do not find peptidoglycan in eukarya. You should know the components of the gram-positive cell wall. Remember, you have a thick layer of peptidoglycan, as well as lipoteichoic and teichoic acids. Compare that to the gram-negative cell wall, where you have a thin layer of peptidoglycan, an outer membrane, and porin structures within the outer membrane, and remember, LPS, or lipopolysaccharide, in the outer membrane as well. Also, can you beat Wicket? What is the dangerous component that's toxic to humans that's found in the outer membrane of gram-negative bacteria? What is that called? That's right, Wicket. As always, it's called lipid A. Remember, lipid A is the lipid portion of lipopolysaccharide, and it's quite toxic to us. So when it gets into our bloodstream, it can cause septic shock and other damage to our tissues. All right, concept check number 3. Which of the following is not found in a gram-negative cell wall? Remember, it's the teichoic acids. Those are found in gram-positive cell walls. But I didn't mention it, but do you remember both gram-positive bacteria and gram-negative bacteria have the periplasmic space? That's the space between the membrane and the cell wall. Next, you should know that the acid-fast bacteria are the bacteria that have mycolic acids in their cell wall. This includes members of Mycobacterium and Nocardia. So these are known as acid-fast bacterium because they have mycolic acids in their cell wall, so they only stain by doing the acid-fast procedure, right? The acid-fast staining method. You should also know that mycoplasma are bacteria that don't have a cell wall. So that means they are pleomorphic. They're not going to have a particular cell shape. You should know that the chromosome inside of a prokaryotic cell is labeled a nucleoid and not a nucleus. And you should know that the nucleoid usually consists of a single circular double-strand DNA molecule. You should know the difference between bacterial ribosomes and eukaryotic ribosomes. Remember, bacterial ribosomes or prokaryotic ribosomes are 70S, which is different than the eukaryotic 80S ribosomes. And you should know that these are found in all cells, and you should know the function is to synthesize proteins. You should know that inclusions and granules are storage areas or storage bodies in prokaryotic cells. You should know that endospores are highly protective defense structures or seeds that can be formed by Clostridium, Bacillus, and even Sporosarcina. Remember that the vegetative cell is the mother cell. It's the metabolically active and growing cell, whereas the endospore is that protective structure that they can form once they become starved of nutrients. Remember that sporulation is the process of forming an endospore, while germination is the process of that endospore growing back out into a vegetative cell. Again, remember, concept checks may appear on the exam. If you discovered that a bacterium you're studying stores phosphate, which of the following would you expect to contain the stored phosphate? Remember, a granule or an inclusion body, right? Those are storage sites. So I would say a granule. Next, you should definitely know the shapes of common bacterium. Coccus shape means spherical. Bacillus means rod shape or capsule shape. Spirillum is helical or twisted rod. Spirochete is like a spring or a corkscrew. Vibrio is gently curved, so sort of like a V shape. Coccobacillus is a short rod. You should know that hyphal cells are filamentous cells. That's what hyphae mean. You should know that pleomorphism is where there is no particular cell shape. And then you should know arrangements. Remember, arrangements and shape are two separate things. Arrangements are how they arrange themselves, how they line up. Do they pair up in twos? That's diplo arrangement. Do they pair up in fours? That's tetrads. Do they form irregular clusters or grape-like clusters? That's staphylo. Do they form chains? That's strepto. Do they form cuboidal packets? That's sarcina. Remember, if it's irregular clusters of cocci, it's staphylococci. If it's chains of cocci, it's streptococci. If it's chains of bacterium end to end, it's streptobacilli. But remember this, if it's bacilli side by side, those are known as palisades. So if the bacilli are side by side, that's a palisade. If they're end to end, that's a strepto. Again, remember, eukaryotic cells are larger than prokaryotic cells, and then viruses are even smaller than that. Remember that viruses are ultra microscopic. That means even smaller than what you can see in a regular lab with a light microscope. All right, concept check number 7. Round bacterial cells growing in irregular clusters would be best described as staphylococci. And do you remember this? I just mentioned this at the end of chapter 1, that archaea, the domain archaea is more closely related to eukarya than to bacteria, and this is because of genetic studies that have been done. So remember that on the exam for sure. Here's another concept check. Organisms in the domain archaea have peptidoglycan in their cell walls. That's false, right? Because think about it, peptidoglycan is unique to bacteria. Awesome. So that leads us to the end of chapter 4. We're about halfway done with this exam review. I hope I'm remembering all the concepts to help you out. I'm doing my best here. But let's take a quick little break time with Gizmo and Wicket, see what they're up to. We'll come back fresh in order to finish off this exam review. All right, welcome back from break time with Gizmo and Wicket. Let's continue on with chapter 5, a survey of eukaryotic cells and microorganisms. You should know the endosymbiont theory. Remember, this endosymbiont theory explains how the nucleus forms. It explains the origin of the mitochondria and the chloroplasts. So I'm going to tell you more about what to remember about the endosymbiont theory. But for now, you should know it's how the mitochondria and the chloroplasts came to be. You should know that eukaryotic flagella have a 9+2 arrangement of microtubules along their length. You should know that eukaryotic flagella beat back and forth or whip back and forth, and that's how they propel the cell. You should know that cilia do not beat back and forth. Instead, they do the power stroke. See, they do a power stroke followed by a recovery stroke. You should know that animal cells do not have a cell wall. However, fungi can have a cell wall, usually of chitin and sometimes cellulose. Algae has all kinds of different cell walls, everything from cellulose, pectin to mannins, silicon dioxide, and even calcium carbonate. So this concept check number 1, if you remember, I didn't like this question, so I'm not going to add that to the exam because this question is kind of misleading. You should know that eukaryotic cells divide by a process known as mitosis, whereas prokaryotic cells divide by a process known as binary fission. You should know that the lysosome is the digestive organelle in a eukaryote. So it's a vesicle full of hydrolytic enzymes for digestion. You should know that phagocytosis is this process of bringing in food particles inside of a vacuole. Do you remember the evidence for the endosymbiont theory? Mitochondria and chloroplasts have a single circularized chromosome with genes that are very similar in sequence to prokaryotic genes, bacterial genes specifically. They have 70S ribosomes as opposed to the eukaryotic 80S ribosomes that they grow on their own and divide on their own by a similar process to binary fission. And all of that suggests that they used to be prokaryotes in the past. Remember, that lends to the endosymbiont theory. You should know that the kingdom fungi, these are eukaryotic organisms that have molds. Molds have hyphal cell shape, and yeasts. Yeasts have roundish or ovid cell shape. You should know that when a mold grows, that interwoven array of hyphae is known as the mycelia or mycelium. You should know that yeasts asexually divide by budding. And remember, fungi are heterotrophs. That means that they feed off of organic material or chemicals. And remember, they have two main modes of nutrition. They are either saprobes, which means they break down organic material to decompose it, or they're parasites. Remember that fungal infections are called mycoses. You should know the difference between sporangiospores and conidiospores or conidia. Remember that the sporangium is a cap in which the sporangiospores are, whereas conidia are exposed, right? The little conidiospores are not within a cap. Remember that that's how you classify fungi, based on the sexual spores that they produce, the zygospore, ascospore, basidiospore. You should know that the zygospore is a sexual spore, whereas the sporangiospores in the sporangium are asexual spores, and the conidiospores in the conidia are asexual. These are asexual spores, while these zygospores, ascospores, and basidiospores are sexual spores. And again, concept check number 5, fungi are generally classified according to their type of sexual spore. Next, remember the algae and the protozoa. These are collectively known as the protists. Algae are photosynthetic, while protozoa are not. Protozoa are animal-like, which means they are heterotrophs. You should know that protozoa are classified based on their locomotor structure or their motility. Are they flagellated, ciliated, pseudopod, right? Or do they have no motility at all? You should know that protozoa have two stages, life stages, the trophozoite motile feeding stage and the cyst dormant stage. Concept check number 7. Which of the following descriptions is true for both algae and protozoa? Remember, only algae are photosynthetic, not the protozoa. They are always unicellular. No, there are many colonial algae, so it's not that. They both contain mitochondria. That's right. It's not they are both heterotrophs because protozoa are heterotrophs, not algae. And then lastly, we talked about the parasitic helminths, the worms, right? You should know these terms. Definitely know that there are flatworms called platyhelminthes, roundworms called nematodes, and then you should know the two types of flatworms. The cestodes, you should know that cestodes are tapeworms, and you should know that trematodes are flukes. You should know that the cestode, the tapeworm, has a scolex head, right? A scolex head with suckers and hooks, as well as proglotted body segments. And concept check number 9, parasitic helminths reproduce by eggs and sperm. Remember, they are multicellular creatures. They can produce the eggs and sperm. All right, and that about leads to the end of chapter 5. Let's go ahead and dive into the final chapter on the exam, chapter 6. All right, are you guys ready for chapter 6? Let's do it. This one has to do with viruses, viroids, prions. Remember that they are obligate intracellular parasites. They are not alive. They are infectious agents. Remember that they can infect all kinds of different organisms. You should know this for sure, that every virus has a nucleocapsid. At the core of every virus is the nucleocapsid. That means a nucleic acid genome as well as a capsid protein coat. Oh, and do you remember that the genome can be DNA or RNA, but not both. And again, we've mentioned this a couple times, but viruses are ultra microscopic. They are so small that they require an electron microscope to see. Remember, not all viruses have an envelope. Some do and some don't. Remember that the capsid protein coat is made up of identical proteins called capsomeres. Remember that there are two forms of typical capsid types or structural types, the helical capsid and the icosahedral capsid. Remember, there are also complex viruses with a complex shape. Remember this, that there are many different variety of genomes that viruses can have. They can have double-strand DNA genomes, single-strand DNA genomes, double-strand RNA genomes, single-strand RNA genomes. And do you remember this? This may be important. Remember that single-strand RNA, if it's positive sense, then it can be translated to protein, right? So you should know that positive sense single-strand RNA is like mRNA in that it can be translated by ribosomes into proteins. However, remember, the negative sense single-strand RNA genomes cannot be translated directly into proteins. They must be converted into positive sense first. Remember that some viruses possess enzymes, and here is a list of different types of enzymes that certain viruses may or may not possess. Remember that these are the steps involved in viral multiplication. And can you beat Wicket? Which of these steps require spike proteins? That's right, Wicket is always adsorption, right? Spike proteins on the surface of the virus are important for adsorbing or binding to specific receptors on host cells. Do you recall that penetration can occur one of two main ways for animal viruses, fusion or endocytosis? Do you remember that enveloped viruses usually exit the cell by budding or exocytosis, while naked viruses usually exit the cell? Remember, exiting the cell is called release, and they do so by lysis or rupturing the cell. So again, let's go to concept check here. Viruses commonly contain both DNA and RNA. That's definitely false because it's either or. You should know that cytopathic effects are cell damage that you can see that occurs to virally infected cells. And you should know that two types of cell damage are inclusion bodies and syncytium. Remember that with animal viruses, you can enter a persistent phase and that these can periodically reactivate. This is known as the chronic latent state. Remember that some viruses are oncoviruses. Remember that oncoviruses are viruses that can cause cancer. So viruses can cause cancer. Viruses can cause cytopathic effects like inclusion bodies and syncytium. Next, remember the bacteriophage. These are viruses that infect bacteria. And here's something important to remember. Bacteriophage do not need to undergo uncoating. Uncoating is not necessary as part of the multiplication cycle of bacteriophage. Remember why? It's because they inject their DNA genome directly into the host. The capsid remains outside. So uncoating is not necessary for bacteriophage, but it is necessary for animal viruses. That's why you see here the multiplication cycle of bacteriophage. Notice how uncoating is missing. And do you remember that many of these bacteriophage have two choices once they infect the cell? They can either enter the lytic cycle, which involves full completion of the viral infection, resulting in lysis and release of virion, or they could enter, remember, a state of lysogeny. This is where the DNA of the virus incorporates itself into the host's genome. Remember that? So that's called lysogeny. So again, the bacteriophage can either enter the lytic cycle, where it explodes and it leaves the cell, or it can enter the lysogenic state, where it incorporates its genome into the host's genome as a prophage. The prophage is when its genome is incorporated into the host. You should also remember that the term induction refers to when the prophage leaves, when the virus leaves lysogeny and releases the prophage from the host genome and then enters the lytic phase. So again, induction can occur, resulting in activation of lysogenic prophage, followed by viral replication and cell lysis. So again, concept check number 5, which of the following is a step found in animal virus multiplication but not in bacteriophage replication? Again, remember that's uncoding. It's not necessary because the virus leaves its capsid outside. Next, remember prion disorders? You should know that prions are misfolded proteins. They are made only of protein, and they're caused by misfolded proteins. And they cause brain damage. You should know they cause transmissible spongiform encephalopathy. And you should know that satellite viruses are dependent on other viruses for replication. Usually these don't have a proper nucleocapsid. And remember viroids? Viroids are simply short pieces of RNA with no protein coat that have only been identified as in plants as plant diseases. All right, concept check number 7, exposure to nucleases that degrade DNA and RNA would damage all of the following except what? Again, prions. Why? Because prions don't have DNA or RNA. So nucleases wouldn't have an effect on prions. Prions are simply misfolded proteins, whereas the rest of these, A, B, D, and E, all have some kind of DNA or RNA to deal with. All right, and that leads us to the end of the exam review. I hope I didn't miss anything. I tried to stress the important concepts to remember. Please study hard, and again, do yourself a favor and don't just rely on this exam review video for everything you need to know for the exam. You know, there is a reason we put together all those handouts and all those individual lecture videos. But I tried to remember the most important concepts to prepare you for the exam. I wish you luck. Do your best, study hard, and let me know how you do.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.